So next, I'd like to welcome our next guest speaker, Dr. Andrew Gascho Landis. Andrew is an associate professor at SUNY Cobleskill. He earned his PhD from Auburn University, a master's from Northern Arizona University, and a bachelor from Goshen College. Prior to moving to Cobleskill, he was a freshwater mussel biologist for the Georgia Department of Natural Resources. Andrew teaches many courses at SUNY Cobleskill, such as watershed and ecological restoration. Here he is talking about freshwater mussel conservation, exploring population trends in the Delaware River Tributaries.
Thank you, Sam. It's really exciting to be here, and I'm really glad to talk about the freshwater mussel work that we've been doing at SUNY Cobleskill over the past several years. So what I want to focus on today is give you a little background information, set the stage, think about conservation of freshwater mussels, and then talk about some changes that we've been observing in the never sink drainage, and then also some distribution issues in the east and west branch of the Delaware and the never sink as well. And then finally I want to end up thinking a little bit about the theme of the conference, the changing environments, and how ecosystem services might play a role in that. All right, so let's jump into some of the conservation issues related with freshwater mussels. I want to start with the global distribution of freshwater mussels. This is the number of species in each region throughout the world, and what should jump out at you is that North America is kind of ahead of the pack with about 300 species of freshwater mussels. All right, so let's take a little closer look at the United States. Really, all of our diversity is in the eastern part of the state. There's really not much going on in the western part of the United States. I'd love to point out Alabama. It's the global epicenter of freshwater mussel diversity. Tennessee's not too far behind, but lots of wonderful species down south. All right, let's scoot up to the northeast. New York, not doing so bad, right? We got about 51 species, and that's much better when we look at the rest of New England that has rather depauperate faunas of only in the teens, right? And so what drives this level of diversity that we have in New York? Well, we're fortunate enough to sort of span three significant biogeographic provinces for freshwater mussels. So we've got a nice portion of the Great Lakes, we've got a little bit in the Ohio Valley, the Mississippi Basin, so that's really the Allegheny River that brings a lot of really great freshwater mussel diversity to the state. And then finally here in the eastern part of the state, we're part of the Atlantic slope drainages, the Hudson, the Delaware, the Susquehanna. Actually have diversities that's pretty similar to the rest of New England, so not quite as many species in this part of the state. All right, I think many of you are aware that mussels are having a hard time, right? So when we look at the conservation status of those 300 species in the United States, 72% of them have some type of conservation, some level of impairment, right? Endangered, threatened, special concern. And so the thing that I really want you to look at here is that only about a quarter of all species of freshwater mussels have populations that are considered stable. All right, so I wanted to look at this same trend for New York. I'd never looked at this data, so I put it together for the meeting. And it's pretty striking that of our 51 species, we have the same level of impairment, almost exactly as what we have at the national level. So 72% of the species have some status. One difference to note here is that species of special concern, and these really are our species of greatest conservation need, make up about half of the species that we have here in New York. And the same, we have about a quarter of our species that have populations that are considered stable, so only a quarter. So why do we have such high levels of imperilment? Well, you all know the story, right? We've treated our streams and rivers really poorly over the last several centuries, and as a result, our aquatic organisms have suffered, especially things like freshwater mussels that are sedentary, right? And they make their living by filtering particles across their gills and respiring using their gills. So any nasty stuff that's in the water, they're extremely vulnerable to that. So even though we've made really great strides in improving our water quality, we're still seeing declines in freshwater mussels. Okay, next thing I want us to jump through, I want to spend some time and just go over the freshwater mussel life cycle. Many of you may be aware of this, but there's a couple things that I want to point out. All right, so we've got our male and female mussel that spend their lives in the sediment. The males broadcast their sperm into the water column. The females passively capture this while they're feeding, and they fertilize the eggs with this sperm. Those eggs turn into glaucidia, which are just larval freshwater mussels. Those glaucidia are brooded in females, and depending on the species, there's one of two different strategies used, either short-term or long-term. Short-term, those mothers may hang on to those glaucidia for only days to weeks after they mature, versus long-term, which could be up to 6 to 8 months after those glaucidia mature. So some different strategies being used here. Once those glaucidia are ready to go, the females start the process of glaucidia transfer. And so this is the really cool part about mussels that I think many of you know, is that they're obligate parasites, these glaucidia, on a vertebrate host, and most often a fish, right? And so these female mussels have evolved a whole broad range of strategies to try to get their babies on a fish, and ideally on the gills, right? So that these juveniles, or I'm sorry, these glaucidia insist on the gills for somewhere between 2 to 3 weeks before they're ready to drop off as juveniles and then start the whole process all over. Okay? So two things that I want to point out about this. First, remember that there are 300 species, so there's lots of different ways that different species have adapted to go through this life cycle. And the other thing is, the more complicated your life cycle, the more opportunities that has to be broken, right? And that it can be broken in multiple places. And so, for example, there's been some really cool research showing that if you have really turbid water, those females have a hard time getting and sorting out the sperm from the rest of the junk that's in the water. Extended cool periods of time can inhibit gonadogenesis and other reproductive processes. You put a dam in the way and those migratory fish can't get to their host, interrupting that cycle. And then finally, these little juveniles are super sensitive to any toxins that are in the water. And so you have poor water quality and these juveniles suffer greatly. Okay, all right, the next thing is I want to talk a little bit about some life history strategies. And if any of you have ever taken a fish ecology class, you were probably required to read Wine, Miller, and Rose. It's a classic paper where they laid out these 3 different life history strategies that they tried to categorize all fish based on things like the longevity of the species, the fecundity, and a suite of different other reproductive strategies that these fish had. This was really successful. People really liked these concepts and these ideas. All right, so the freshwater mussel people were pretty much 20 years behind on this. And in 2012, Wendell Hegg published his book, and he laid out how he thought these concepts of life history strategies, these 3 different categories, applied to freshwater mussels. And so he really spent pretty much most of his career amassing the data on different species characteristics and traits so that he can actually start to categorize them into these different classes. And so this is, to give you an idea of what this looks like, for freshwater mussels we'll have these opportunistic species, and they have really low lifespans, right? 8 to 10 years is all the longer that they live. They grow really fast. They tend to have thin shells and a low age of maturity. Sometimes even the age zeros are reproducing. I've seen it happen. On the other end of the spectrum, you have these equilibrium species that they have these long lifespans, right? Minimum ages of 30 years typically, can be up to 80 or 100 years in some cases. They're going to grow pretty slow, and they're going to have a delayed age of maturity, and their fecundity is relatively low. All right, then we have these periodic species that fall kind of right here in the middle between the ends of this spectrum. And so it's been fascinating for me over these past 10 years to see how much this concept has been embraced by freshwater mussel biologists. And it's been used quite a bit to help explain the patterns and trends that we've seen in freshwater mussel populations, right? And so that's a little bit what I'm going to do today. I'm going to use this as a lens to look at some of the issues that we're having with freshwater mussels. All right, so let me jump into the first study that we looked at here on the changing freshwater mussel communities in the Neversink River. And so the impetus was, this funding was provided by the Brook Floater Working Group. And the goal was to do a range-wide exploration of what the species status was. This was run by the USGS co-op unit out of UMass. And so we decided that the Neversink had historically been a stronghold for brook floaters in New York, and that it would be worthwhile for us to get back in there and actually figure out what the status of brook floater is. The cool thing for us is, while we're out there, we can collect all the mussels in the community and enumerate them and kind of figure out what the current status of those are. So we did that in 19 and 20, and this is a picture of Mike DeMontpied. He was a student that did this for his senior research thesis. He helped me out with this. All right, so the Neversink River, it's in southeastern New York. It is a tributary directly to the Delaware River. The upper parts of the watershed are drinking watershed for the New York City, and so you couldn't ask for like a better protected watershed in the headwaters. It's highly forested. It has a hyperlimnetic reservoir on it that helps control water levels and for this system. Then in the middle we have the Neversink unique area. And I bring these things up because you couldn't ask for a better system. It's got really great protection in it. And so if we would expect mussels to be doing well anywhere in the state, it would be a system like this. Okay, so we started off, we did field surveys, and we followed the Brook Floater Rapid Assessment Protocol, which really requires 2 hours of search time per site. And so the way that we did this, we would have 4 searchers look for 30 minutes. These were independent surveys, and we did this by, we all had our own 1-meter lane, and we would travel up the river looking for mussels. And so when they're independent, it means that you wouldn't sneak over and try to steal mussels out of your neighbor's lane, right? And we did this because we wanted to be able to use an occupancy approach to look at some of this data. When we were done with our lane, we actually measured the distance traveled so we know how far we actually went and how far we surveyed. We ended up doing or collecting data across 56 sites in the Neversink River. We did collect a suite of habitat variables. I'm not going to talk about those today. All right, so the historic change part of this, I had a lot of people that came before me that did really great work, starting with David Strayer in 1990. So he did the first surveys in the Neversink River, the first system-wide surveys. Great original data. Then the lovely Barry Baldico came along in the late 90s and did another round of surveys. In 2008, Heather Galbraith out of USGS, the former Wellsboro lab, did a bunch of surveys really over 3 years surrounding 2008, and then I came along, right? And so we've got this really nice 30-year set of data that we can start, excuse me, assessing the trends. And really that's all I want to show you today is through time, what's happened to abundance and diversity in this system. Okay, I want to start with these are the species we caught, right? There are 7 different species in this system. We were excited because we were able to capture all of the species that were historically present in this system. So they're all still there. I will say some of them are at really low numbers. Like for example, I only caught 1 alewife floater in the system. But anyway, we were able to they're all still there. Okay, so looking at abundances and how that's changed through time, we've got our years down here on the bottom from the 1990s in the past to the present, and CPUE is really just the number of mussels that we caught in 1 hour. And so this relationship is interesting, well, because there is no relationship. So really it appears that there has been no change in the abundance of freshwater mussels in the Neversink through time, and we still had some sites in this river where we had really great catch rates. It was really exciting to work in these areas. All right, so now this is where the story gets a little sad, right? In that when we look at diversity using Shannon's diversity index, we see these massive declines in diversity, such that in the past, in the 90s, some of these sites had really great measures of diversity, right? And then in the 2000s, this really kind of collapsed. So the next thing that I want to do is to go back to our life history trace, right? And take our 7 species that we have and get them into the different categories where they fit. And so we end up having 2 opportunistic species, 4 periodic species, and just 1 equilibrium species. So our equilibrium species is the eastern elliptio. On the opportunistic end, the thing I want to say about is I'm actually not going to look at those anymore because this is like the alewife floater. There's only really a few of the eastern floaters. So I'm really just going to focus on showing you a couple graphs of the periodic species and what's happening with those. The other thing I want to say about the periodic species, 2 of these are species of special concern for the state of New York, and the other is federally endangered. Okay, so when we look at, and so how I want to look at periodic species is just what is the proportion of the catch that's a periodic species, and how has that changed through time? Right, and so this looks very much like that diversity graph, in that in the 90s, periodic species made up a really large proportion of the catches in the river. And then now, they make up almost none of our catch, right? And so the flip side of this result is that eastern Elliptio is making up almost the entire catch, right? That's the mussel that we're getting when we go out there. The last thing I wanted to do with these periodic species is to think about the distribution across the river. I wanted to look at that historic data especially to see, are there places that those periodic species were more abundant or more present in the river? And so to do that, we split up the Never Sink into 9 different sections. Part of the reason that we did that is because we didn't all sample in exactly the same place. So we wanted to come up with a reasonable way to split the river up and combine data to look at these different segments through, from the top of the river to the bottom down near the Delaware. And so when we did that, here are the data. So the black circles are the pre-2000, the 1990s data combined, and the open circles are the 2 surveys in the 2000s. And so starting here on the right, this is up by the dam, segments 8 and 9. And so historically, there really weren't any periodic species that were located up by the dam. What's interesting, though, is that these middle sections of the river is where we had really high proportions of periodic species in the 1990s. And then, right as you get closer to the Delaware River, those dropped off. And that's actually kind of a normal pattern that we see in freshwater mussels, is that those periodic species tend to get replaced by the equilibrium species as the systems get larger. Okay, and then of course here, essentially those periodic species are disappearing in these middle sections of the Never Sink River. All right, so I'm going to go back to think about some life history strategies and stuff just a little bit more, because these different life histories relate to the types of environments where you find these animals, right? For example, opportunistic species tend to be in disturbed, stressful habitats, right? So for freshwater mussels, this might be a sandbar that's shifting habitat. But these species can get in there, grow fast, have lots of babies, and colonize those areas. All right, the other end of the spectrum are the equilibrium species. They're really requiring stable habitats, right? And when they are in these stable habitats, they can develop really large population size. And that's what our eastern Elliptio is doing, right? It has these really large population size. It's enjoying something about this environment that's stable. Okay, in the middle, we have our periodic species that like some level of intermediate disturbance, right? And they have these, they tend to thrive in these environments that have this cyclical environmental variation. And they're really well known for having highly variable annual recruitment, right? So that there's some cue that happens in the system and they have extremely high levels of recruitment and then maybe none for several years after that. All right, so something seems to be going on that our periodic species are not happy, they're not doing well. Something in their environment appears to have changed, right? And so we're not finding very many. And so when my student techs find one, they're actually really excited about it and want to get their picture taken with these periodic species because there's not very many of them. They're really proud to find them. All right, so what has changed? We tried to wrap our heads around what in the system has changed between the 90s and the 2000s in this environment that maybe make it unsuitable for this type, these periodic species. And one of the things that we came up with is the flexible flow management plan, right? And so this is a plan that applies to all 3 of the drinking watersheds in the Delaware system, and this was initiated in 2007. So this is a quote, it explains it, comes off the DEC website, and it says, generally, the flexible flow management plan generally uses higher base releases to promote wildlife habitat rather than rely on fixed storage banks for temporary relief from adverse conditions. So to sum that up is we're going to release more water all the time instead of episodically when we might think we need it, right? Especially in periods of low water, right? Okay, so my student Mike thought this was really fascinating, and he wanted to take a look at how that actually changed flows in the river. And so he went to the USGS gauge right there at the Neversink Reservoir and calculated the monthly averages for the 20 years prior to 2000 and the 20 years after 2000. And what they said would happen actually happened, right? We have much higher flows currently, this yellow, mustard yellow line up on top here, than we did historically, right? And so what really strikes me about this is especially this summer. During the summer when we would have periods of lower flows, now we tend to have at least 2 to 3 times higher levels of discharge during the summer months. Okay, so these periodic species really appear to be collapsing in the Neversink River. And I guess this is my opinion that flow seems to be playing an important role in this, right? There's something has changed in their environment and flow has changed, and that might very well be connected. The other thing that we know, though, is that this is a hypolimnetic reservoir on this system. So when we add more water to the system, we're adding more cold water to the system, and lots of it, right? So to follow up on this and to think a little bit more about temperature, we were really excited to do some follow-up surveys in 2023, this past year. Lisa helped us get this started, and again, the focus was on brook floaters. We wanted to look for where some rare species were at. And so there were some historic records, especially from the East Branch of the Delaware, so we set up a bunch of surveys in the Eastern West Branch. We were excited about this because now we could take a little bit of a deeper dive into looking at the effects of temperature on the freshwater mussels in 3 systems instead of just 1. All right, so we continue to use our brook floater rapid assessment for searching for these things. One thing that's a little different about the Neversink, and maybe I didn't say this clearly enough, is that we didn't actually survey above the reservoir at all. All of our surveys were below the reservoir, but in the East and the West Branch, we did surveys above and below. And when we were surveying below, we wanted to make sure we had a really nice gradient from the dam basically down to the confluence in Hancock for both of those systems. So we had a nice representation of that system to see how those water temperatures changed and how the mussel communities responded. Okay, so the next thing that I needed here, and for the East and West Branch, was some temperature data. And so kind of a shout out to Tim Picorni, pointed us in the direction of EcoSheds, some great temperature that's online. And so thanks to the Region 4 folks that helped make sure this data from the East and West Branch get online here. So what I did, just as kind of a core start here, is I wanted to use at my sites monthly average temperatures. And so I went in and got data from these loggers, right? And I paired that with the distance from the dam, so I could develop a relationship of distance from the dam and what the temperature looked like, as in this case, let's say just average August temperature. So I went in and got the last 4 years, average August temperature from the last 4 years. It allowed me to make these really nice, simple, linear regressions so that I could predict at my sites what those average August temperatures might look like. I did the same process for the Neversink, but in this case I used some temperature data from the USGS Wellsboro lab. They had some nice temperature data for me to do this. So this is what this looks like. These are the really nice, simple linear regressions that I was able to make here. As we get further from the reservoir, this is the logger data. We've got really great relationships. So I felt pretty confident that I could predict at my sampling locations, right, below the reservoir, what those average August temperatures were. Okay, so again, I'm going to start with the catch. I actually put numbers on here for you to see these. These are just below the reservoir on the East Branch. And so we had 1,000 elliptio in our survey sites, but only 8 of these other 3 species, right? So I'm not going to really get into it with the periodic species on this part of the story, but maybe you can tell that they're not doing very well in this system either, right? Because of such low catches that we had of these animals in this system. All right, so now I'm going to look at our temperature data impact on abundance. I'm going to start with the West Branch here. We have abundance on the y-axis and our predicted average August temperature on the x, and we found no mussels in the West Branch. And this was above or below, and this is just from below, right? But we found no mussels anywhere in this entire system. Okay, looking at the East Branch, we were really kind of excited to find some really nice populations once temperatures got to about 20 degrees C as this average August temperature. All right, now adding the never sink to the bottom here, a little messier, right? Because I had a whole lot more sample sites on the never sink as compared to the East and West Branch. But one of the things that starts to pop out is once things get to an average August temperature of about 15 degrees, we actually start seeing mussels enter into these river systems, right? And it's really not until at least in the never sink, until they get between 20 and 25, this average August temperature, that we see really robust populations of freshwater mussels. I also added on your distance below dam, so looking at this same data, for those of you that might be interested in like, well, how far is that down below the dam till things warm up? And I think the thing to point out, the difference with what's going on with the West Branch, is that it's only 18 miles long before it hits the confluence. So that's not long enough for it to actually warm up to support freshwater mussels, right? Versus the East Branch, that's about 32 miles long till it hits the confluence. So it is long enough to warm up enough. It also has the added benefit of the beaver gill dumping some additional warmer water into it. And then finally, with the never sink here, same thing. It's really not until we get about 35 miles from the reservoir we see really robust populations. We have a little bit of a blip on here at around 15 to 20 miles, and this is again the effect of a tributary that's dumping some warm water into this system that helps support some freshwater mussels in that part of the system. Okay, so then now I just want to take a quick look at richness. This is just number of species. Same thing, and right, so we've got increasing richness as we get to these warmer temperatures. And so what this is really suggesting is that it's these periodic species, right? They are really sensitive to these cold waters. And when we have one species, it's always the eastern liptail. And these additional species, we really don't see them until temperatures are getting closer to 20. The never sink's a little different. Again, I think this is the effect of that tributary there coming in. We see a few additional species, this first little spike of species, but it's really not until we get to about 20 degrees that we really see species diversity kick up in these systems. I just wanted to put this one graph in here because I've been working with Dan Stish and Suni Oniata to work on some occupancy model stuff, the way that this data was set up to be analyzed. We're starting to see so we really just started this process. So this is really kind of a future directions or next step part of this. But we see some striking results from the never sink and the East Branch that the pattern is the same, right? From the graphs that I just showed you, that our probability of occupancy, once temperatures get up to 20 degrees, it's 100%, almost, right? That they're going to be there if the water's warm enough. If it's not, you're probably not going to find many mussels. Okay, so I wanted to show you this one just to show some of the upstream numbers for freshwater mussels and temperatures. And so this is just from the East Branch, and if you look, kind of those black dots in the upper left-hand side of this graph, that's the temperature. So there was some temperature loggers up there. Thank you, TU, I think is the ones that manage these temperature loggers. And what we see there is that those temperatures above stream are about 20 degrees on these average August temperatures. But then it takes approximately 50 miles, right, until temperatures get back to about 20 degrees in August, and that's actually when we see mussels come back, right? We had mussels above, and then finally see some mussels below when average August temperatures get up to 20 degrees. I showed this to a student, and they said, so really there should be mussels over that entire 50 miles? It's like, well, probably, right? Okay, so let's think about so it seems pretty clear that temperature is limiting the distribution of freshwater mussels below these dams, right? So how does temperature impact freshwater mussels? So I put up our little shot of their life cycle here. And a couple ways that I think temperature could be limiting their distribution is Well, A, it just slows their metabolism. It's like they're living in a perpetual winter state. And so they have limited energy stores. They probably have reduced reproductive investment so that they're not able to reproduce. The other thing that's probably going on is that they're lacking the thermal reproductive cues that they need, right? And so there was a paper that just came out in 2019, great paper that showed that Elliptio complanata, right, the eastern elliptio, it releases Lochidia at temperatures between 16 and 19. So if your average August temperature, which should be the warmest time of the year, doesn't get above 15, you're not able to reproduce, right? And so this is for the hardiest species that we had out there. And so this is, I think, a pretty clear explanation as why we're not seeing Elliptio complanata in temperatures less than 15 degrees, or at least not very many. Well, we maybe find just a very, very small number of them. Okay, so then I want to, for my last few minutes here, I want us to think a little bit about the theme of the meeting here and this idea of changing environment, right? And this can mean lots of different things. I think at first blush it might be, oh, well, climate's changing, that's a changing environment that we have to work with. But actually one of the first things I thought it was is really the changing human environment that we're working in, right? And how people and society is changing. And I guess I'd like to think that society is changing on how it's valuing the environment, right? And so it's more common that people are thinking about issues related to biodiversity. We're starting to value things like ecosystem health, right? So this is good. And so the last thing that I wanted to do is just a little bit of a thought exercise for myself, right? Because freshwater mussels they're often touted as nature's water purifiers, right? And so I thought, okay, this could be pretty interesting. I want to figure out what are the ecosystem services that were not being realized, that are lost because these cold temperatures are inhibiting freshwater mussels from living in the area, in these sections of the river, right? So I just want to step you through quickly how I did this, right? So what I wanted to do was estimate how many mussels I think are missing from these systems and what the filtration rates are so that we can figure out what the lost ecosystem services are. Okay, so I'm doing this for the East Branch below the dam. And so what I did is I just determined the average number of mussels per meter squared from site, right? So we had our 1 meter lane, we knew how long we went, I could average those across surveyors to get a pretty nice average density per meter square at each site. I multiplied that by stream width to say this is how many I would estimate to be in this 1 meter cross section of river. I did that for multiple sections along the river at sites where I thought temperature wasn't playing a too negative role for these mussels. All right, so this is what this looks like as I just developed a really simple relationship between these sites with mussels, and I did this for every meter downstream of the dam. So I estimated every meter downstream of the dam how many mussels I think were there. And in the East Branch, I came up with 9.5 million mussels that could be living downstream of the dam. The next thing I did was actually use the data or catch data to figure out how many mussels I think are there. And these are just simple linear regressions to